friends, and welcome to our 6th video in Beginners Bioinformatics series. Let's learn some more fundamentals. In this tutorial, we will go through some basic information about protein structures, a few softwares used to predict protein secondary and tertiary structures. We will also learn by example how to retrieve data from primary protein databases. Let's begin with some fundamentals. Genes are specific regions of DNA which possess codes to direct the synthesis of proteins. This code begins with a start codon and ends with a stop codon, and the DNA in between constitutes an open reading frame or ORF. Genes transcribe and translate into a chain of amino acids which constitute the primary structure of proteins, represented as a series of single-letter amino acid codes in a protein database. This primary structure folds into alpha helix and beta pleated sheet, secondary structures, which further fold into a tertiary structure, or sometime into a quaternary structure. Organic molecules with a central carbon atom, an amino group, a carboxyl group, a hydrogen atom, and a side chain, which is unique for each of the 20 essential amino acids. This unique side chain makes an amino acid to have a particular physicochemical property like hydrophobicity. 20 essential amino acids can be grouped based on their charge, polarity, and hydrophobicity. Primary structure is formed when two amino acids join by peptide bonds, eliminating a molecule of water. This long chain of amino acid, or primary structure, has an amino end and a carboxyl end. In bioinformatics, we write primary structure as a series of amino acids which are stored in databases like Protein Database in NCBI and SwissProt and Tremble databases in UniProt Knowledge Base or UniProt KB. This is the homepage of Protein Database from NCBI, where sequences have been collected from several sources. We can type the name and species of an organism, and data, if available, will be displayed. Here, by typing insulin protein sequence for Homo sapiens displays many options, and by choosing one appropriate option and by clicking on this hyperlink, the annotation page is displayed. The size of this protein is 110 amino acid, and the accession number is AAA 59172.1. We can find other details as we scroll down the page. The FASTA sequence is displayed through FASTA hyperlink. We can copy this accession number to use it later for UniProt KB search. SwissProt and Tremble are another two important protein databases which are part of UniProt knowledge base. SwissProt has entries which are reviewed by a team of researchers, whereas Tremble has computationally generated sequences translated from the EMBL nucleotide database. We can either search UniProt by typing the name of the protein and organism, which here shows 1086 records for reviewed or SwissProt entries and 527 records for unreviewed entries from Tremble database. If we use the same accession number we took from Protein Database in NCBI, we will be shown the same insulin protein entry here. However, the SwissProt annotations are more detailed, as we can see here. Protein secondary structures, alpha helix and beta strands, are formed due to coiling or folding of the primary structure. These structures are stabilized by multiple hydrogen bonds. In bioinformatics, we can predict how a given primary protein sequence is going to fold as an alpha helix, beta strand, secondary structures by using prediction tools or softwares. There are many such softwares available like JPRED and PSIPRED. In JPRED, we input a protein sequence in FASTA format and make predictions. It may show in PDB, but you may continue to predict. Here are the results: green arrows for beta sheets and red tubes for alpha coils. For PSI PRED, choose the box, input the sequence. Results show yellow color for beta strands and pink for alpha helices. These predictions are based on algorithms such as neural networks. Proteins acquire tertiary structure by further folding of the secondary structures, stabilized by disulfide bonds, van der Waals forces, hydrogen bonds, and some ionic interactions. A successful three-dimensional folding is very crucial for protein functioning, as proteins failing to fold properly can lead to diseases like Alzheimer's, Parkinson's, Huntington's, or cystic fibrosis, etc. Shows protein and nucleic acid three-dimensional structures is Protein Data Bank or PDB. Structure database in NCBI also stores protein three-dimensional structures. These structures are obtained by techniques like X-ray crystallography and NMR, which is nuclear magnetic resonance, or can be generated by homology modeling in silico. You can input the name of the protein or PDF entry number, and if structure has been deduced for this protein, it will be displayed. For example, here we see a part of the estrogen receptor protein. Several protein three-dimensional structure prediction softwares or tools such as FIRE, Swiss Model, ECPRED 3D are available, which can build a 3D model of the given primary sequence. FIRE 2 is a protein homology or analogy recognition engine which uses the hidden Markov models for the alignments. Swiss Model is an automated comparative protein modeling server. EasyPrep 3D is an automated homology modeling program based on neural networks. This is the home page of FIRE2. We input the FASTA format sequence of a protein in the given box. Here we are using estrogen receptor beta protein sequence and predict the tertiary structure of the protein. Choose normal or intensive modeling mode and press FIRE2 search. The results take a few hours, so you must provide your email address and have patience. This is the normal mode model for liver X nuclear receptor beta, which is similar to human estrogen receptor beta. Here 66% sequence of human estrogen receptor beta has been used to create the 3D protein model with 100% confidence. Using intensive mode, FIRE2 chooses multiple templates from all the available hits, and here we can see 6 models all predicted with 100% confidence. Swiss Model is another software to predict three-dimensional structure. You can start modeling the 3D structure by inputting your sequence and click on Build Model at right-hand side, where you can change the structure to any of the model like space fill or ball and stick models. You can even rotate it to study its overall three-dimensional structure from all sides. There are several other features which can be explored for required use. One important use of node the three-dimensional structure of a protein is for studying its interactions with the drug molecules. This figure from PDB shows a human serum albumin protein bound to warfarin drug. Finally, we look at the quaternary structure of a protein, which is found in multi-subunit or oligomeric proteins. For example, in hemoglobin, LDH or lactate dehydrogenase, glycogen phosphate molecules. These structures have 2 or more protein subunits held together by ionic and hydrogen bonds, disulfide bonds, and weak hydrophobic forces. That's all for this tutorial, friends. Hope you understood the demonstrations. If you have any questions and suggestions, please comment. Your comments are always welcome. If you have liked this video, please like and subscribe to my channel to keep me going. In this series, our last video will be on phylogenetics basis. Don't forget to watch. Thank you and See you soon.